Hello everyone. Greetings for the day. Thanks to the organizer for giving me the opportunity and inviting me to give the talk, which is use of human fetal brain cells for neurotoxic study, which includes human brain cells, how it has been used for HIV mediated TAT protein as well as for Parkinson disease and Alzheimer's disease. As we know, a range of neurological disorders are causing serious impairment to nervous system, and these neurological disorders are caused mainly due to irreversible damage to the neuronal cells. And the animal models so far used are not realistically represents the human disease, and the disease results obtained using this animal model and those research also often fail to hold true in human setting. In such a scenario, use of human brain cells culture system as an in vitro system is a reasonable approach. As we know, human brain is the most complex organ of our body. In fact, it is the in charge of our entire body. It deals with consciousness, ideas, feelings, concerns with the intelligence, memory and thoughts. It functions with highest cognitive order. And we know we can see here different parts of the brain has a specific function. And most importantly, cerebral cortex is responsible for thinking, reasoning, speaking and understanding what we think. If we compare our brain with other mammals, we can see here the brain to body mass, when it increases, the mammals have greater performance, and increasing cortical folding and surface area of brain is correlated with the complex behavior of human. And we can see the development of the human brain takes about 40 weeks. To know more about the comparison of the rodent brain and human brain development, we can see here the initial phase of development such as neurulation, migration, neurogenesis are more or less same for the rodents as well as for human. But the latter processes such as the afferent efferent neuronal corrections, synaptic for synapse formation, and the behavioral pattern establishment are more complex in human being. And more importantly, we can see here by fMRI study, it has been shown that basics of neural systems are established by 10 weeks of gestation, and all the structure continue to develop throughout the fetal period and up to the early childhood. By 6 years of age, brain has reached 90% of the adult volume, and by age of 25, typically the brain is fully developed. And here I want to mention that we have used the fetal brain of the week gestation week 6 to 16 age of gestation for the fetal brain cell isolation. Now, why these human fetal brain cells are important? Because the human fetal brain cells or these neural precursor cell system is a powerful experimental tool, and it differentiates into the neuron, astrocyte or oligodendrocytes based upon the specific media. And this NPC models offers a research window to understand the normal disease state and toxicity study of the human brain. And this in vitro model provides continuous supply of rare human brain derived neural precursor cells as well as the differentiated cells, and which can be cryopreserved and revived as per the experimental need without depending upon the fresh human brain samples. We can see here the culture procedure, how we have done the human fetal brain cells with the approval of a human ethics committee as well as the approval from the Indian Medical Council of Research. We have taken the brain from the civil hospital Gurgaon, and the brain was dissected aseptically, and they are cultured in the PDL coated culture flask for a period of 5 to 7 days, and then those cells were passaged and identified by their morphology as well as characterized by the immunospecific markers. We can see here, this is a subventricular zone of human fetal brain from where the cells were taken out, and those cells we can see here, they are expressing nestin, the neuronal stem cell marker, and this is overlaid by the DAPI, which is a nuclear staining. To see here, we have taken the brain cells from the subventricular zone, and in the figure A, you can see the patch of tissue, brain tissue have attached, and that tissue pieces grows over the period of 2 weeks to attain the huge size in day 14, and from that day 14, the cells, neural stem cells, has sprout out and become the monolayer cells, as we can see here. And then we characterize these neural stem cells to know the specific marker protein expressed, which is the nestin. The most important nestin is the neural stem cell marker, and there is no expression of the GFAP, which is astrocyte marker, and absolutely no expression of the TOJ1, which is neuronal marker. To further characterize, we have used these markers, which are Ki67, MUSA-C, and SOX2. They are expressed in these neuronal precursor cells. And also to point out here, these monolayer cells can form the spheroid, which is called neurospheres, and they can grow over a period of time by size. And when we stain them with the neural stem cell marker, it expresses nestin, SOX2, as well as Ki67, as like monolayer cells. And when we differentiate these human neurospheres, it also expresses nestin as well as the neuronal marker, which is TOJ1, because they are in the process of differentiation. In the lower panel, you can see the expression of the nestin as well as TOJ, and G-stack image showing the sections of the neurospheres. When we started the differentiation of the neural precursor cells towards the astrocytic lineage as well as the neuronal lineage, it took around 2 to 3 weeks, and the end of the 3rd week, we can see here the astrocytic marker expression GFAP has increased, as well as in the neuronal marker has increased by the period of 3 weeks. And initial days, very less expression comes, and at the end of the day, end of the 3rd week, we can see the TOJ1 expression is almost all the neurons. And here to mention that these differentiations are based upon the specific medium given to the neural precursor cells. To characterize the human neurons, we have used the tyrosine hydrolase, which expressed in these neurons, MAP2, synaptophysin, as well as dopamine transporters are expressed in these neurons. To characterize the astrocyte, we have used S100 beta as well as GFAP, and we can see the overlay here along with the nucleus DAPI, which is shown in the blue color. Also, to further characterize, we have used RT-PCR as well as Western blotting to see the expression pattern of mRNA as well as protein, which increases over the period of 3 weeks from day 0 to day 21. We can see the increasing of TOJ1 during the neuronal differentiation, and the GFAP expression goes on towards the astrocytic differentiation. Now we come to the human neural toxicity used by the TAT protein, and why we have used this TAT protein, because HIV, we know there are several subtypes, and in Western countries subtype B is predominant, whereas in India we have subtype C, and we have in India there are less neurotoxic observations has been found. So our interest to use the TAT protein of both the subtype and see if there is a difference in the neurotoxic pattern. And we know the mechanism of HIV pathogenesis is either a direct pathway by secreting the chemokine and the cytokine from the astrocytic cells or the non-neural cells, and or by direct pathogenesis to the neuron by the viral protein. So we went ahead and used the TAT protein where we can see the neurotoxic regions which starts from 31 to 61 position of the amino acids, and we use the TAT protein of virus B as well as virus C to compare both the viral proteins to check the neurotoxicity. And our experimental design, we can see here, we have directly treated the viral protein to the neuron as well as indirectly treated to the astrocyte and collected those supernatant to check the toxicity of neuron by in situ tunnel staining, MTT assay, Caspase-3 isolate, Caspase-3 activation, and also by looking at the oxidative stress level and the mitochondrial membrane depyrylation level. We can see here, this is a tunnel assay has been done by a dose dependent study of the TAT protein. We can see here the TAT B is causing more cell death, which is depicted by the red staining, which is comparatively more than the TAT C protein. And also when we see the MTT assay, which is also a cell death assay, TAT B causes more cell death. We can see here in the figure A, that means the cell viability goes down, which is comparatively less than the TAT C. And in the right panel, we see the production of the MCP1, which is a cytotoxic protein, which is secreted more in TAT B than the TAT C. And also when we see the oxidative stress, TAT B causes more oxidative stress than the TAT C. And when we do the mitochondrial depyrylation assay, we can see here TAT B creates more aggregates of green colors. Why we see this? Because JC1 probe gives the green color when there are more mitochondrial depolarization. So this figure indicates that TAT B causes more mitochondrial depolarizations. And the TAT B also causes the dendritic degradement. So the development of the dendrite is being impaired by the TAT B, which is more impairment causing than the TAT C. To summarize this part of the HIV TAT mediated neurotoxicity, we find that functional difference in TAT B, which is clearly visible from the TAT C in terms of damage to the human neuron, oxidative stress, dendritic arborescence, depolation of mitochondrial membrane potential, as well as the production of cytokines MCP1. So now we come to the mechanism of the Parkinson disease, how this neural precursor cells has been used for this study. We know Parkinson disease is a progressive neurodegenerative disease and characterized by the degeneration of dopaminergic neurons in the substantial nigria pars compacta, and activation of the mixed lineage kinase and the C-Jun N-terminal kinase, that is Janus kinase, has been reported in the model of Parkinson disease. In this study, we use p38 inhibitors that protect primary dopaminergic neurons from MPP mediated cell death and prevent the downstream phosphorylation of p38, p53 and its translocation to the nucleus in the ventral midbrain. As we can see here, p38 inhibitors confer the protection against MPP mediated cell death, and this selective, when we use selective inhibitors, we can see in the panel A and B the cell death by MPP has been attenuated, has been declined by using the P38 inhibitor. And in the right panel C and D, we can see the localization of the protein P38 has not been gone to nucleus, and it was inhibited. So the cell death has less, and it protects the neuron, which we call that it preserves the surviving of the neuron. In another study of the beta amyloid toxicity in the human neuron, as we know, amyloid plaques and neurofibrillary tangles are the hallmark of Alzheimer's disease. And this beta amyloid is a primary component of the amyloid plaque, and it is neurotoxic. In this study, using human neuron, we show that tetrahydrocurcumin, which is THC, which is a metabolite of curcumin, shows a protective effect against the oligomeric beta. We can see here co-treatment with THC reduced the number of tunnel positive cells in the left panel, which is quantitatively shown here, and in the right panel, THC reduces the reactive oxygen species level as well as decreases in the mitochondrial membrane potential as well as the caspase-3 activity. To summarize, the in vitro study finding correlates with the clinical observations made in AIDS dementia patient in India, where there is low prevalence in HIV-associated dementia. And using those Parkinson model and A-beta model in Alzheimer's, we have seen that considerable attention has been directed towards identifying compounds which are having neuroprotective property. Not only us, there are several current studies across the globe is going on, which is done by various international team of scientists, and this human stem cell based model has been created by various researchers, and this is one of the example where the brain organoids has been developed from neural precursor cells. And in the right panel we can see oligodendrocytes can be formed from the neural precursor cells, and myelin has been developed, myelin secreted neurons has been developed from these oligodendroid cells. So these works which I presented has been done at National Brain Research Center, which is highly acknowledged. Thank you.